Hi, my name is Ashlyn Huntley and I'm going to be doing activities 2 through 3 in the Aseptic transfer lab. I have my sanitized workstation that I cleaned with a 10% bleach solution, my disposal jar with 90% isopropyl alcohol, my PPE, and right here I have my culture that has been growing for 3 days. I have my fresh nutrient broth, my agar slant, my agar plate, my paraffin candle, and my inoculating loops. I have labeled the plate, the slant, and the fresh nutrient broth with the culture name and the date. So first off, I'm going to grab my culture, completely close the cap, and mix the contents. I'm going to remove a sterile inoculating loop from the package. I'm removing the cap of the reactivated culture. And flaming the mouth of the candle, or mouth of the tube. I'm going to insert the inoculating loop and get some of the culture. Flame the mouth of the tube again. And replace the cap. My fresh nutrient broth right here. I am going to remove the cap, flame the mouth, insert the inoculating loop into the broth and introduce the culture. Remove the inoculating loop, flame the mouth, dispose, and replace the cap. I close it tight to 100%, and now I'm reopening about 50%. Next, I will be doing the transfer from the broth culture to the slant. I'm going to remove a sterile inoculating loop from the packaging. I'm going to remove the cap of the culture, flame the mouth, insert the inoculating loop, flame the mouth again, replace the cap, now I'm grabbing the agar slant. I am removing the cap, flaming the mouth, I'm holding the tube at an angle and inserting the inoculating loop while touching the bottom of the agar. I'm drawing the loop back in a zigzag motion. I have not made any dents in the agar. I'm flaming the mouth of the tube. Replacing the cap. Putting that in the disposal jar. I've closed this 100% and I'm reopening it to 50% and setting it down. Now I am doing the transfer from the broth culture to the plate. I am removing the cap of the culture, or I'm grabbing the inoculating loop first. inflaming the mouth of the tube. I'm inserting the inoculating loop and getting some of the culture. I'm inflaming the mouth. Replacing the cap. I'm going to lift the petri dish slightly, not completely. I'm used to shield from any contamination in the air. I'm dragging the loop gently onto the surface of the agar in a back and forth motion. Close the lid, place the loop in the jar, and I'm going to let that set for a minute before I invert it. And now I'm going to incubate all 3 cultures for 48 hours, then take a picture, then 72 hours, and see where we are there. Right now I'm going to start disposal, and we'll see how it goes in 48 hours.